Hello everyone, welcome to Tax Solution. Today we are presenting a brief discussion on chromosome, its structure, its location and its functions also. Now what is chromosome? Chromosome, the microscopic thread-like part of the cell that carries hereditary information in the form of genes. A chromosome is a DNA molecule, deoxyribonucleic acid, with part or all of the genetic material of an organism. Chromosomes are normally visible under a light microscope, but only when the cell is undergoing the metaphase of cell division, where all chromosomes are aligned in the center of the cell in their condensed form. This compactness of any chromosome is a defining feature. For example, the 46 chromosomes found in human cells have a combined length of 200 nanometer. If the chromosomes were to be unraveled, the genetic material they contain would measure roughly 6.5 feet in length. This compactness of DNA molecule prevent it from becoming an unmanageable tangle. DNA wraps around proteins called histones to form units known as nucleosomes. These units condense into chromatin fiber, which condense further to form a chromosome. This is called nucleosomal model of chromosome. We will be discussing about it in our next presentation, so stay tuned with us for these updates. Now, this compactness of chromosomes play an important role in helping to organize genetic material during cell division and enabling it to fit inside structures such as nucleus of a cell. The average diameter of a nucleus is about 5 to 10 micrometer. In animal cells, chromosomes reach their highest compaction level in anaphase of cell division. The structure and the location of chromosomes may vary between prokaryotes, viruses, and eukaryotes. The prokaryotes such as bacteria and archaea typically have a single circular chromosome, but many variations may exist. Prokaryotic chromosomes enclosed within a nuclear membrane and have less sequence-based structure than eukaryotes. Bacteria typically have a point from which replication starts. This point is called the origin of replication, whereas some archaea contain multiple replication origins. On the other hand, the non-living viruses have chromosomes consisting of either DNA, deoxyribonucleic acid, or RNA, ribonucleic acid. This material is very tightly packed into their viral head. Now, in eukaryotes, the chromosomes are contained in a membrane-bound cell nucleus. Chromosomes of a eukaryotic cell consist primarily of DNA attached to a protein core, but they also contain RNA. Chromosomes in eukaryotes are composed of chromatin fiber. Chromatin fibers contains into a structure called chromatin. Chromatin allows the very long DNA molecules to fit into the cell nucleus. During cell division, chromatin condenses further to form microscopically visible chromosomes. The structure of chromosomes varies through the cell cycle. During cellular division, chromosomes are replicated, divided, passed successfully to their daughter cell to ensure the genetic diversity. Now we are going to discuss about the general structure and parts of a chromosome. There are 6 main parts of a chromosome. They are pellicle and matrix, chromatids, centromeres, secondary constriction site, satellite, and telomere. Here, a labeled diagram of chromosome at anaphase stage of cell division. Now the first part of chromosome is pellicle and matrix. Pellicle is the outermost covering of the chromosome, formed of non-genetic material, and inside the pellicle a jelly-like substance called matrix. It is also composed of non-genetic material. The matrix is formed from the nucleus during telophase of cell division, but the structure of matrix and pellicle has not been supported by several workers like Darlington. He discarded the presence of pellicle, and the existence of matrix and pellicle has not been supported by electron microscope also. Now, the second part of chromosome is chromatid, chromonema, and chromomeres. The chromosomes are made up of chromatin. The structure of the chromosome is best studied in the late prophase, metaphase, and anaphase. During early prophase, a chromosome appears to consist spirally coiled thin or filamentous long continuous structure called chromonema. In plural, this is called chromonemata. In this structure, you can see this. The chromonema thread is marked at intervals in a linear order by a number of knot or granular or beaded-like structure arranged vertically in a single row along its length called chromomeres. These chromomeres are clear and small dense-like structure at fixed intervals along the total length of chromonema thread and appears like similar to the beads on a string. The number and position of chromomere is found relatively constant in a chromosome, on basis of which a chromosome can be differentiated from the other chromosomes. These chromomeres are responsible to carry the genes during inheritance. In the late mitotic prophase, the chromosomes appear as thick, dark, and various shape and sizes. Under electron microscope, chromosomes at metaphase stage are visible clearly as a double structure consisting of two chromonemata, which become intertwined with each other. Has its own set of chromomeres and is known as chromatid. Thus, there are two chromatids in a chromosome. The two chromatids are joined together at a point along their length, which is said as centromere or primary constriction or kinetochore, the third part of the chromosome. As we said in our previous slide, the third main part of chromosome is centromere. The centromere is clear, non-stainable part of the chromosome. The centromere has a significant role in nuclear division. It performs two roles, that is holding the two chromatids and binding the spindle fibers. The position of centromere is constant for a particular chromosome. This point also plays an important role in chromosomal movement during mitosis. The structure and the function of the centromere is different from that of the rest of chromosome. During division, the centromere is functional, while the rest part of the chromosome is genetically inactive. In mitosis, metaphasic chromosomes which consist of two chromatids, 4 granules may be seen within the centromere. These granules are called centromeric chromosomes. During anaphase, when the two chromatids separate, each chromatid shows 2 granules. At the same time, duplication of the centromere occurs during mitosis and meiosis. The centromere is considered to be an extended region of the chromonema. Now, depending upon the position of centromere and comparative length of two arms of chromosome, chromosomes may be following types, such as acrocentric, submetacentric, metacentric, and telocentric. Now, what is acrocentric chromosome? Acrocentric chromosomes look like English alphabet I, rod-like chromosome having one arm of very small size. Thus, arms are unequal. Position of centromere is subterminal for acrocentric chromosome. Now, what is submetacentric? Submetacentric chromosomes look like English alphabet L or J. For submetacentric chromosome, centromere is not present in the center of the chromosome. Arms are unequal. Now, what is metacentric chromosome? For metacentric chromosome, centromere is present in the center of the chromosome. For that reason, this chromosome is look like English alphabet letter V. Arms are equal in length. Now, what is telocentric chromosome? Telocentric chromosome having a terminal centromere. For that reason, it look like English alphabet I, and it is a single-armed chromosome. On the basis of number of centromeres, the chromosomes may be divided following types, such as acentric, monocentric, dicentric, and polycentric. Now, what is acentric? The chromosome which is without centromere is called acentric. When the chromosomes have only one centromere, it is called monomeric. When the chromosome with two centromeres is termed as dicentric. Now, what is polycentric? A chromosome possessing more than two centromeres is called polycentric. Now, what is secondary constriction area? Secondary constrictions are the constricted or narrow region found at any point of the chromosome other than that of the centromere, which is primary constriction site. The difference between the two constrictions can be noticed during anaphase, as chromosomes can only bend at the site of primary constriction. The secondary constrictions are useful in identifying a chromosome from a set. There are either 0, 1, 2, 3 or 4 secondary constriction sites in a cell at anaphase. Some parts of these constrictions indicate sites of nucleolus formation and are called nucleolar organizing regions or NORs. The nucleolus in the nucleus remains associated with the NOR of the secondary constriction site. The chromosome may have a rounded or elongated body at its end. The rounded body is connected by a thin chromatic filament. It is called a satellite. In diameter, it is equal to or smaller than that of the chromosome on which it is present. Satellite-bearing chromosomes are called sat chromosome. Sign acido thymonucleonico. Now, what is telomere? The chromosomal terminations at either end are known as telomeres. They have specific uniqueness. A broken end of an chromosome can fuse with that of the other chromosome, but intact parts of the telomeres can't fuse in any case. Because of this type of polarity, the telomere prevents the other chromosomal parts to fuse with them. Now, we can see in this diagram chromosome showing secondary constriction site, telomere, primary constriction sites, satellites, and chromomeres, chromonema. This is a model chromosome structure based on light microscopic studies. And here in this below figure, you can see the satellite region also. Now we are going to discussing about some characteristics of chromosome. There are two types of chromosome, sex chromosome and autosomes. Autosomes control the inheritance of all the characteristics except the sex-linked ones, which are controlled by the sex chromosome. Humans have 22 pairs of autosomes and 1 pair of sex chromosome. Eukaryotic species has a characteristic number of chromosomes, which is called chromosome number. In species that reproduce asexually, the chromosome number is the same in all the cells of the organism. Among sexually reproducing organisms, the number of the chromosomes in the body, means somatic cells, is diploid, a pair of each chromosome, and the haploid number found in the sex cells or gametes. The haploid number is produced during meiosis. During fertilization, two gametes combine to produce a zygote, single cell with a diploid set of chromosomes. Somatic cells reproduce by dividing, a process called mitosis. Between cell divisions, the chromosome exists in an uncoiled state, producing a diffuse mass of genetic material known as chromatin. The uncoiling of chromosomes enables DNA synthesis to begin. During this phase, DNA duplicates itself in preparation of cell division. After replication, the DNA condenses into chromosomes. At this point, each chromosome actually consists of a set of duplicate chromatids that are held together by the centromere. The centromere is the point of attachment of the kinetochore, a protein structure which is connected to the spindle fibers. During the middle stage of the cell division, the centromere duplicates and the chromatid pair separates. Each chromatid becomes a separate chromosome at this point. The cell divides and both of the daughter cells have a complete set of chromosomes. Chromosomes uncoil in the new cells again, forming the diffuse network of chromatin, and the process going on. Now, what are the functions of chromosome? Chromosome can control the physiological behavior of an organism with the help of the genes which are present in them. We know each chromosome is made up of DNA, and this DNA by replication gives rise to messenger RNA, which carries the genetic information in form of code. The mRNA comes out of the nuclear wall into the cytoplasm, where it helps to form a particular kind of protein needed by the cell or the body. Chromosomes are a hereditary vehicle carrying genetic information from one generation to the other. Now that's all for today. If you like our content, then don't forget to subscribe our channel. For more updates, you can follow our Facebook page also. If you like this content, then please let us know your valuable feedback in our comment section, and please like and share our content. Thank you. Have a good day.